Okay. All right, let's get started. Let's see, where do I start? Are you waiting for your grades or you don't care? You want them? Okay. The assignments are coming this week, the essay most probably next week, the exam next year. I'm slowly getting there, but should be there soon. So I got the first question about the new essay, so that's a good sign. That means there's one person reading it. Anybody read it? Anybody start reading it? No? You too? Okay. Tell me what's different. So the class realizes and wakes up. What's different about this?
It's more empty.
It's more.
It's not like the first article was like we did this, and this.
Mm-hmm.
This is more.
Okay. Anything to add?
It's like a lot of things we don't know about, I guess, because we never learned it in molecular biology, so
Yeah.
new things, I guess.
Okay, so let me tell you about this paper. It's different from the other one. It's not a research paper, kind of not a research paper. It's more of a computational paper, right? And it's closer to a review than to a research article, an original research article. And so you'll see that the methods are kind of everywhere. This team did that, they got this, we did this, they got that. Okay, that's the first part. Second part, is it easier to understand or harder?
It's a bit harder,
Harder? Really?
because like they talk about things that you have no idea.
Okay, right. So, okay, maybe it's a bit harder because it has more technical stuff that are unrelated to what you learned before, but very close to genomics. So most probably you'll have to talk about those a little bit more than before, right? Because most people don't know about the computational stuff that were done and the techniques that were used to kind of figure out the genome of the Neanderthal. So that's the first thing. Second, it looks more like a compilation of stuff. Instead of step by step, we did this, we did that. And so it gives you a little bit more open-ended essay that you can write. Obviously, you can take it anywhere you want, but the idea is now to kind of summarize that paper, okay? More summarize than actually talk about what they did and limitations and whatnot. So instead, you're going to talk about just the paper, what's in it, and expand on stuff that seem to be a bit complicated to understand for you guys, okay? So it's a simpler assignment, although it looks not as simple, because it's kind of open-ended. You can do whatever you want with it, but the idea is to summarize it in 4 to 5 pages, okay? I'll let you guys read it, wait for your questions, and then I'll tell you exactly what's happening, okay? This way it's going to be a little bit easier. What else? Did you start working on the presentations? No? Too much work? Too much work. Okay, it's not bad. It's not bad. It should be quite easy. You can read the paper and then just present the figures. Typically, you present the figures to give a little bit of background, the methods used, the figures, and that's it. Okay. No need to show all the figures. You can show 2/3 of them or whatever. That just depends on how big your paper is. If your paper, so some students send me a review paper. So those I don't really like because you could basically just reread the paper to me and that just doesn't tell me if you understand or not. While a research paper actually requires you to think before you can talk about it, right? So this is why I want the research articles. For the team that has the single-cell RNA stuff, let me get back to you, okay? Because I have to make sure that it's of the appropriate level, okay? What else? Who didn't send me their paper? What's happening? Okay, I'll be waiting for your paper. So we're almost done with the semester. There's 3 more boring lectures, and then 4 interesting ones, and then I stop working, okay? And then it's you guys who are presenting, so it should go pretty fast. You have very little amount of assignments that are happening right now, so you can focus a little bit more on your other classes. I already given you way too many, so this should be good. Just one thing, don't wait until the end of the semester to start studying, because the 4 talks that I will be given will be included in the exam: gene therapy, microbiome, prostate cancer, Neanderthal. So those are very important to focus on, and they're going to be somewhat big. So it's important to kind of do them one by one, so that you don't get to the final and then you have to read the paper and understand it and then ask me questions and how, okay? With that, let's talk about selfish DNA. So at the beginning of the semester, if you remember this slide, I showed you all the pieces of the eukaryotic genome. We talked a little bit about genes, centromeres, replication origins, telomeres, and with genes we talked about regulatory sequences, but what we didn't mention is we talked a little bit about selfish DNA, if you remember the TY transposon in yeast. Today we'll take a deeper dive into selfish DNA, and then we'll also talk about functional RNAs in the next 2 classes, and then from there, the cool topics. So the human genome, if you remember this slide also, I was talking about the composition of the human genome, and you can see approximately 45% of the human genome is transposons. So this is a major part of the genome, and as you go through the complexity of the genome, you will see this portion here increase, right? So obviously yeast had a lot less compared to humans, and there are also other sequences that are kind of selfish, which are repeats. If you remember, we talked about the microsatellite, mini-satellite, and all that. So these are the 3 types of repeat sequences that exist in the human genome. Some of them, they occur, so basically these transposons and tandem repeats, they replicate themselves, or they go through cell division and all that in a selfish way. They have no real purpose in the cell, okay? That's at least what they were before. Now we're realizing that they can actually cause mutations and things like this, so this idea is a little bit changing. And these repeated sequences can happen within genes, right? Some of them are the genes, for example, gene duplication, right? Homologs, paralogs, orthologs. So these duplications are repeated sequences in the genome. Sometimes you have 11 repeats of the same gene. And then you have these tandem repeats, which we were talking about, those markers that are used to map different genes, right? So the microsatellites, mini-satellites, etc. Okay? Today we'll talk about the transposons. Tandem repeats, I mentioned them a little bit previously. They're really boring, so there's not much to talk about other than the last 2 slides which talk about them. And the duplications we already mentioned with the homologs, paralogs, and orthologs. Okay? You guys saw transposons before, right? You know a little bit about this. So transposons are just DNA sequences that move from place to place within the genome. They actually are able to what we call they kind of jump from one spot to the other. Sometimes they land in a gene, sometimes they land outside of a gene, and they tend to have no role. So they do replicate themselves, but the end goal is nothing. If it ends up in a sequence that is useless, like jumps from one sequence to the other, and there's no change in that sequence or whatever, then there's no purpose really other than replicating itself, right? As I said, 45% of the human genome is made up of transposons. This is crazy. There's more transposons than genes, obviously. They're found throughout the genomes. Usually the size is between 50 base pairs to 100 to 10,000 bases. And you can have, depending on which type of transposon, so there's many families, you could have 1 copy to several hundred thousands of copies depending on the family, okay? There are 2 types. The major one that is being actually active at this moment right now in your body is the retrotransposons. The DNA transposons were usually active, but more in the past, so basically earlier during evolution. And the difference between them is that one uses a copy and paste mechanism, so it requires a reverse transcriptase, while the DNA transposon is just a cut and paste mechanism. And this is just, when we talk about these mechanisms, we're talking about how one transposon can go from one spot to the other, okay? For the retrotransposon, you need to copy it and then paste it somewhere else, while the DNA transposon, you need to cut it directly from your genomic DNA and then insert it somewhere else. There are two types, subtypes to these types. So there are the autonomous type and non-autonomous type. Autonomous means that the transposon itself encodes all the proteins that are required for its translocation, while the non-autonomous type will depend on the machinery of another transposon. Okay? So for example, a non-autonomous retrotransposon will usually use the reverse transcriptase from another transposon to translocate from one spot to another in the genome. Okay? If there are DNA transposons, then instead of using the reverse transcriptase, they will use, for example, integrase or transposase or whatever, or the endonuclease even. Okay? Simple? Okay, so very simple way to explain this. The retrotransposons, you can imagine your DNA, right? It has a sequence in it that is the, let's say, the retrotransposon cassette, and it contains genes that are important for this transposition. When we're talking about retrotransposons, the DNA sequence in your genome will actually be copied into RNA. That RNA molecule will then be reverse transcribed into DNA, and that DNA will be integrated somewhere else in the genome. Okay? So you can see that retrotransposons have the ability to copy themselves, and you'll see that DNA transposons don't, okay? Because they cut and paste somewhere else. What else? They're very similar to retroviruses. As a matter of fact, you'll see that some of the genes are found in retroviruses, like the envelope gene, which makes the envelope of retrovirus. But in the case of retrotransposons, it has no use, right? So you can think of it as being a, let's say, originating from some sort of viral infection at some point, okay? What happens when retrotransposons are inserted into the DNA at a different locus, you'll have your endonuclease that cuts the DNA, and because it cuts it, it creates a staggered end, not a blunt end which is straight, a staggered end. And that staggered end will bind to the retrotransposon. Now, when that happens, you leave a short piece of DNA that is empty, right? Because you're basically cutting your retrotransposon, you're cutting your genomic DNA, and then you're pasting them together. When you paste them together, you will leave sometimes these overhangs, and these overhangs will be repaired by the cell machinery to repair DNA. That will create these sequences which are kind of duplications called target site duplications. Okay? So wherever you find these target site duplications, you know that there is a retrotransposon that was inserted there most of the time. So this is something that is known from them. This is how they are studied. As I just mentioned, we talked about the TY element in yeast. You can take a look at the earlier slides. So for the retrotransposon, there are three types. There's the long terminal repeat transposons, which resemble retroviruses. You can think of HIV, you can think of whatever other retrovirus you know. They have these long terminal repeats, and these long terminal repeats, they allow for the positioning of the transposon whenever it moves. Okay? So these terminal repeats kind of dictate the site where the transposon will be binding, will be inserting. The LTR transposons have a pole gene, and this pole gene contains produces two proteins, the reverse transcriptase and the endonuclease. Okay. Now the other two types, they don't have an LTR flanking the transposon on each side. Instead, they're just your regular DNA molecules, And the way they insert in DNA is kind of not clear. It depends really on the integrase that integrates them in the DNA. So the integrase is somewhat specific to, depending on which integrase is being used, it's specific for a certain sequence, okay? So there are two types of these retrotransposons without the LTR. One of them is called the lines, the other is sines. Lines is short for long interspersed nuclear elements, and the other one, obviously, it's short interspersed nuclear elements. The difference between them is the fact that one of them has the genes that encode for the reverse transcriptase, endonuclease, and the RNA binding protein. And then the other one does not have the reverse transcriptase gene, and so what happens is that it will use it for its own purpose, okay? So the sines are non-autonomous, the lines are autonomous, and the LTR are autonomous as well, okay? Is this clear? Easy, simple? Okay, let's dive a little bit deeper. So this is how what they look like. So retrotransposons, as I told you, there are two categories: LTR, retrotransposons, and non-LTR. The LTR, they look like this. You have these long terminal repeats on each side. You have the TSD, or target site duplications, that occur from the insertion. And then you have these genes, GAG, GAG, and ENV. If you look at any virus, you'll see that they have these. Well, most viruses, they have these sequences, so you can think of it as being, it came from a viral infection at some point. The pol gene also exists in viruses, and this one here, for instance, codes for two proteins, the reverse transcriptase and the endonuclease, which helps in cutting, okay? So in the DNA, you have long terminal repeats. These long terminal repeats will be recognized, and the endonuclease will cut there and then insert these new transposons that are copied through a step of RNA, cDNA insertion, okay? Is this clear? Maybe, kind of, okay. If it's not, you can ask me. So the other type, they don't have the long terminal repeats. So this is your typical transcript. So you have your line and sign. Let's take a look at the line first. Long interspersed, what the hell was it called? Jeez. Nuclear elements, sorry. And so the line, they have the shape of just your normal RNA molecule. Well, it's a DNA molecule, but then gets converted into an RNA molecule. You have your poly A tail at the end, okay? And so the mechanism through which they replicate is a little bit different. They still require reverse transcriptase and endonuclease, but they know when to stop based on the poly A tail, okay? Not based on this stuff here, the LTR. So in this case, they don't have any cool name. It's ORF1 and ORF2. ORF2 is the gene of the line element that contains the endonuclease and reverse transcriptase, okay? Sign is kind of like the line, but without the genes, right? And there are two elements, specific elements that we'll be actually looking at, and those are the most frequent elements that are found in the human genome. For the line, we'll be looking at the L1 element, and for the sign, we'll be looking at the ALU element. And we'll see why these are important and how come they're studied as part of the genome structure. Questions before I jump? No? Okay. So the line 1 element is exactly how you saw it right here. It's the same thing. So you have the ORF1, which goes for P40, but forget about that. And then you have the ORF2, which has the endonuclease that cuts, the reverse transcriptase, and this very important poly A tail, which I'll talk about in a second. What's kind of interesting and a little bit weird compared to the line is the sign, and we're looking at a specific example called the ALU repeat element. So the ALU repeat element produces these two monomers that are kind of, their origin comes from Actually, I will explain this in a second. I don't want to mix you up, but just know that the structure of these ALU has two sequences that code for a left monomer and a right monomer, which are about 30 base pairs. Okay. The ALU element is about 300. Actually, I'm going to talk about it now. I'm trying to go faster than the slides. Okay, L1 element. So the L1 element, 17% of the human genome is basically L1 elements. They're each 6 kb long, so you can do the multiplication to figure out how big is our genome. It's about 3 billion bases, and there's about 6,000 full-length copies of this L1 element in the genome. Okay, the rest are not full copies. There's about 500,000 L1 elements in DNA. So this is a crazy amount. They're one of the major parts of our genome. Now, I have a question for you before we move forward. How come do we have very similar genomes and we're able to do a consensus genome sequence if we have about 45% of the DNA being transposons and they can jump from place to place? Shit. Yeah.
This is just a gamble, but the idea is that even if you track jumping from place to place, if you do it across multiple copies of the genome, you'll notice that the jumping is not always the same. And if you make a model of all the various genome copies together, you can map something that is cohesive across all genomes and map the entire genome.
That would give you, though, a sequence that is kind of shuffled, right? So it would be like this one is before this one, but we don't know where exactly it is compared to the others, right? So, yeah.
Delete all of the L1 elements when you think about your consensus sequence?
That is a very good idea, but we have them in the map. Should have asked it in the exam. Maria, yes.
Maybe they can only go so far, so they only jump across.
Very good point, but no. Yes, Gabriel.
It says there are only 80 to 100 are active, so maybe it's not all 6,000 that are jumping around. It doesn't look like that much.
Yeah. Okay, let me write the names, then I'll give you the explanation. But basically, obviously the cells are not going to let this happen, right? So we're evolving, right? So the cells, what are they going to do? They're going to create these repression mechanisms to prevent them from jumping everywhere. So we'll talk about this in the end of the slides, but these are one of the main causes for human mutations, okay? In fact, the L1 insertion into different genes will lead to 1 in 1,200 human mutations. So every 1,200 human mutations that happen in the genome, one of them is caused by an L1 insertion. And every 1 to 50 individuals, in 50 individuals will carry a new insertion that does not exist in other people, okay? So this counts for a lot of the variability within the human genome and one of the major causes of many diseases, okay? So let me just write the names. Yeah, I think that's okay. So there's that. The ALU element, which is a little bit more interesting, is about 11% of the human genome. So together, 28% of the genome. This ALU element is about 300 bases long, and it's derived from an RNA, the 7SL RNA, which is part of the signal recognition protein particle. Signal recognition particle, and basically the 7SL RNA resembles the sequence. So we know where this ALU element is coming from. And the role of the 7SL RNA, unrelated to the course, is just to tell you the origin about the ALU element, because you would technically ask, why the hell do we have this stuff in our DNA? So this signal recognition particle is a protein complex that when a polypeptide is being translated and then we detect a piece of it that is a signal peptide, the SRP will bind to it and then stop the translation. And the region that recognizes the polypeptide is the 7SL RNA. Okay? Unrelated to the course, just some evolutionary biology kind of piece. What we're more interested in is the fact that this ALU element, there's 1.1 million copies in the genome. So every 3 kb, 3,000 bases in the human genome, we have an ALU element. And this ALU element is the cause of more than 20 human diseases. Okay? So, second question. Actually, I will ask it in a second, but you know what, why not? I'll ask you here. So the ALU element seems to be causing a lot of the human diseases, but it's less present than the L1 element. How come? Jeez. Yeah.
It's less present because it's shorter, but there's more places where it inserts, so it's more likely to insert somewhere that causes the problem.
Yep. So very simple answer. 6 kb is much more harder to translocate than 300 bases. And so these guys jump a lot more. That's pretty much it. Okay. There's a little bit less of it, but it's more spaced out evenly in the human genome, and because it is more evenly spaced out, then there's more chances that it jumps to some sort of gene compared to the L1 element. Okay. The other thing is the fact that there's not many of these that are active, while this guy here, most of them are active. Okay. So this is the big difference. So the ALU element is not autonomous. It requires the reverse transcriptase of the L1 element. And so if there's no L1 element, these guys cannot do their job. But they don't need anything else other than this reverse transcriptase, because this sequence here is transcribed normally. Okay? So this is basically transcribed using an RNA polymerase. Very simple. This is one of the other reasons why it is causing a lot of problems. It's because it does not require anything special to be translocated other than the reverse transcriptase. Now, 1 in 30 individuals have a new ALU element insertion. So you can imagine how most of our genes are actually messed up by these things. Not messed up. Sometimes they actually have a evolutionary advantage to their translocation. So this is why I put selfish DNA, because actually it's not that selfish anymore. We understand it as one of the mechanisms through which evolution happens. Okay? If, for example, this moves to a gene and breaks it, and that gene is causing you a health problem, then this is an evolutionary advantage, right? If it takes with it an exon, and we'll see this at the end of the lecture, if it takes with it an exon that suddenly goes into a new protein, then you're giving that new protein a piece of the other protein, and so you're making this chimeric protein that could have a new function that is advantageous for you. Okay? So these guys are jumping as we speak. The ALU elements. So I'm going to talk a little bit about evolution of the ALU elements just quickly to give you a perspective of why they're interesting. First of all, the ALU elements are becoming less and less active. And the reason for that is that they're acquiring these new mutations in them. So DNA, as it replicates, it leads to random mutations happening. And so you get them being less active. Obviously, you also have the opposite scenario where you make it more active. And that is dependent on this poly A tail at the end of the ALU element. Because the poly A tail is the piece of DNA that tells you where to stop transcribing. Okay? So if you're reverse transcribing, this is very important for your activity. If you have a bigger poly A tail, then you have more chances of stopping the transcription, reverse transcribing, and moving, other than compared to these pieces here where you have mutations that actually can make you miss it. Okay? What else? Now, these ALU elements, when we talk about them in genomics, they're often linked to primates. And the reason for that is that they're found in high numbers in primates, and they're used to actually figure out which primates came first, when we're talking about evolution, right? And so you can see here that each of the primate has its own signature of ALU elements. So if you just take a random DNA molecule and sequence it, then you'll be able to tell which one it is because of these ALU elements, okay? And obviously, with time, there were some deletions, insertions, things like this. And so there are changes in the ALU set, the set of ALU elements that are specific to each one of the organisms. Actually, these are the ones that are used to map primates. Okay. What else? So the other thing is the ALU elements, they tend to mutate because every time you have a transcription and then reverse transcription step, you are prone to mutations. But it seems like these mutations, they don't affect their translocation. The reason for that is they depend on reverse transcriptase from somewhere else. And so because they depend on reverse transcriptase from the other L1 element, then it doesn't matter what happens to its sequence. It will still move around. The only thing that is affecting the ALU element is really how the poly A tail is mutated. Okay? Other than that, there isn't much that can affect its translocation. One interesting thing, though, about the polymorphisms is the fact that each group from different parts of the world have their own specific set of ALU mutations. So this is what is shown in this slide here. And then depending on the group, you can actually take a look at these ALU polymorphisms and figure out if the person is North African, East European, or whatever. Okay? So you can read on this. Questions? No? Maybe? Going fast here. Okay. Yeah, should be good. So I just asked you a question of how come the ALU elements, the transposons are not jumping everywhere and breaking the DNA, and we have a consensus genome that is similar between each person. And so how come it's not different due to these jumping? Well, it's because the cells have grown a mechanism that is able to prevent, to shut down transposition, right? Because if you let it happen, within a day or two, you basically be dead. Let's say you start transposing from now, your DNA will be destroyed and that's it, okay? So one of the most known, not known, but the most numerous family of proteins is called the Crab zinc finger repressor proteins. And these are regulating proteins for these transposons, okay? So we have about 400 copies, and this has doubled from the divergence of primates. So we need more variety in these crab zinc finger repressor proteins because we have more ALU elements and mutated ALU elements and L1 elements, okay? Zinc finger, if you remember, it binds DNA, right? So zinc finger, I told you about it a few times. This means that these proteins bind to DNA, and they will probably recruit a set of proteins, okay? Yeah, so here what I wrote here is that these proteins can act to suppress retrotransposition. So it's a bit weird because initially they were found to bind to regions of DNA transposons and then prevent their cut and paste mechanism. But in this case here, they seem to be able to also suppress retrotransposition, which depends on transcription, and so it's a bit weird how they're able to do it. One thing that we understand about this is the fact that the crab zinc finger, there is many of them. Each one is specific to a DNA sequence, and this DNA sequence will be recognized by this crab into an open chromatin structure. So if there is open chromatin, there is a chance for cutting or for retrotransposition. And so what you want is you want to prevent that, right? And you do that by having these crab proteins that recognize this DNA sequence and recruit this protein complex, which has the CAP1 protein. This is an inhibitory protein. We don't care about all this stuff here. I just want to mention something. So crab zinc finger recruits CAP1. CAP1 recruits the NERD complex. The NERD complex, you'll see a lot of it in most of your classes because the NERD complex is important for methylation and deacetylation of DNA. And so what it will do is that it will close or open the DNA based on which of the HDACs are found in it, okay? And so what happens is that when you have this crab zinc finger protein binding to DNA, then that means that this piece is vulnerable to transposition, and you want to prevent that. And you do that by either closing up the DNA or changing the structure of DNA through this NERD complex. Easy? Okay. So just a few examples and a little bit of evolution biology. Zinc finger 91, so crab zinc finger 91, blocks transposition of SVA retrotransposon. Zinc finger 93 is the L1 element. So if you want to play with the L1 elements, you just break this zinc finger here and you'll allow for transposition of these elements. And these ones are only found in primates. They evolved, obviously. This is what this slide is saying. So, for example, about 8 to 12 million years ago, the zinc finger 91 gained some features that allow it to inhibit retrotransposition. Okay, boringly enough, well, I mean, it's a little bit boring for me, but maybe not you guys. When you read a lot about the retrotransposons, you're mostly reading about their evolution. Not much is known about their disease-causing mechanisms other than the fact that they insert inside other genes, okay? What else? So, you can read this if you're interested about this, but basically these two have evolved through time, and this is just because what happens is the cell will prevent the transposon from kind of being selfish, and then the transposon will gain mutations that will make it evade this new mechanism that is trying to shut it down, and then it just keeps going on. So this is why I called that slide intergenomic arms race, because they're basically both fighting, and then all you end up with is a higher amount of these crab proteins and better mechanisms through which these transposons jump from one place to the other. What else? Okay. Now, what we're discovering now is a little bit more interesting. So retrotransposons seem to be regulating genes. And this is a bit weird. It's kind of not understood very well. But one of the retrotransposon families, the LTR5HS, LTR, so now you know what type it is. And then just family 5, and this is the name of the retrotransposon, which was active about 15 million years ago, is still conserved. So if it's conserved, that means there were some evolutionary forces that kind of kept it conserved through time. As you can see, the others are evolving nonstop, and they're fighting with the shutdown mechanism to try to evade it. But this specific retrotransposon family seems to have stayed conserved across time. And one team, I can maybe send you the article about this, did some work using CRISPR-I, which now you know what it is, and then you can guess what CRISPR-A is. And they basically tested this on the retrotransposon, and they found that when you break this retrotransposon, you end up affecting the expression of other genes that are far from the transposon. As a matter of fact, in the cells, they found that it activated about 1,800, or let's say 1,800 genes, and repressed about 143 genes without affecting 275. Cool. Okay, I think we have time. So we talked about this branch, we talked about this branch. Now there's another branch that is the DNA transposon. So this one here does not require reverse transcriptase. It's more of a cut and paste mechanism, so it requires an endonuclease and an integrase to integrate this piece of DNA somewhere else. Okay. The DNA transposon is the cause for this variegated maize or corn colors here. And the reason for that is because of its insertion into different pigments in the DNA, okay? So depending on where it inserted into that specific little piece, then it will change the color, okay? So we'll go back a little bit to bacterial DNA transposons. So the transposons were first discovered, the DNA transposons were first discovered, obviously, in bacteria. Anything you discover is related to antibiotic resistance. So when these guys moved from one spot to the other, they either gave resistance or not, because the antibiotic resistant gene was inside the transposon. And then you had these inverted repeats on the opposite sides of the transposon. So this is kind of the structure. These inverted repeats usually contain a protein called transposase, and transposase just will transpose the transposon from one spot to the other, okay? And it does so based on these inverted repeats. So it finds these inverted repeats somewhere else and then places that piece of DNA in there. There are two of them that are very well known that you guys know also. If you ever heard about tetracycline resistance and then kanamycin resistance, well, they were discovered from these transposons. The transposon number 10, which is flanked by the insert sequence number 10, has the TET resistance gene in the middle of it, and the TN5, so transposon 5, with the inverted sequence 5, has the kanamycin resistance gene in it. Obviously, when they were studying these, if you became resistant to these antibiotics, that means you were probably you probably had this transposon. If not, then you don't have it, okay? Humans. Humans, very similar. For humans, we have these inverted terminal repeats. So instead of going this way, they're going this way. And they're kind of similar to the long terminal repeats. The only difference is that the long terminal repeats go in the same direction. That's the first thing. And then they encode a nuclease, which is called transposase. This nuclease just cuts and pastes somewhere else. And usually what these DNA transposons use is this part here to kind of move from one place to the other. Obviously, some DNA transposons, they lack the transposase, so they're non-autonomous, and they require to use the transposase from another transposon. So DNA transposons are a little bit older in terms of evolution compared to retrotransposons. There's only about 3% of the genome that is DNA transposons, but most of them are inactive. There's about 125 families in the human genome, and that gives you a total of 100,000 elements. So there's a little bit less, about 1/5 of the L1 element. Okay. They were very active in the past. Primate radiation here just means when the kind of different groups of primates emerged. So when you're talking about primate radiation, you're talking about this. This is primate radiation. Okay. Wait 1 second. So their activity ended about 37 million years ago. I don't know how they get these numbers, but probably some computational methods, because it's quite hard to figure out what the hell was there before. Obviously, if you have samples and all that, you can compare them, but maybe I can talk about this next lecture. You know, it's very complex to figure out 37 million years ago what was happening, but I guess they have some computational methods to predict these numbers. So I told you that these DNA transposons existed about 37 million years ago. They were very active during primate radiation, but then they kind of stopped being active. So what happened? Any idea? No?
It's a question in natural history. What happened? There was some kind of big evolutionary change somewhere.
Yeah.
Something to do with single cell organisms.
Yep. So anybody else want to add, or?
Yeah, does it have something to do with bacteria?
Actually, not sure for the bacteria. The idea about the question is more like if you're able to think of a mechanism that kind of shut them down because they were pretty much useless. So these guys move from one place to the other, and so at some point the cells probably just grew some mechanisms to shut them down, similar to the crab kind of mechanism. But it's not really clear what actually happened. It's just suddenly they stopped being active, and they're actively researched. And we're trying to understand why the hell, well, not me, but other researchers, to understand why they stopped being active. Okay. What happened is that there are mechanisms that shut them down, but it doesn't seem like they are acquiring the ability to evade this transposition. Most probably because of the lack of transcription in the mechanism. Okay, so if you were to test your hypothesis, you can try to find these proteins in the cell. Do you guys have any idea how you can do this? Come on, guys. Want me to help you a little bit? So we learned about sequencing. Transposons move. So let's start about that. About there. So if you sequence the sequence of a person for 1 million times every day, let's say, you'll see that they are moving or not, right?
Yeah, and a certain percentage of the time.
Sorry?
A certain percentage of the time. There's a
Yep.
movement occurs and other things.
Yep. So basically the way you would see if there is there is a mechanism that stops them from moving or whatnot, first of all, you need to find them where they are moving. And you do that by sequencing the genome, because that will tell you that this piece of DNA moved from here to there. And then from there, it's finding the protein. And to find the protein, you need to figure out the proteins that are bound to the DNA, right? Crab. Days you guys are sleeping, yeah? Need a little bit of partying. Want me to wake you up? Pop quiz. No. Okay. It's okay. We're almost done. We're almost done. The less evolutionarily boring piece of the lecture, okay? So transposons, they have these mechanisms where they affect gene expression. And they have caused a lot of the genomic change throughout history. And they do this by having, there's 3 mechanisms, mainly the insertional mutagenesis. So the transposon moves from one piece of DNA to the other. And because it inserts inside of a gene, it can inactivate it, or it can make it worse. Usually it's inactivation because it's way too big to actually add something interesting to it. It can also cause, if you remember, we talked about the different duplication mechanisms, whole genome, gene, segmental. So segmental duplication is caused by the movement of these transposons. And the way this happens is that recombination usually requires both sides of the recombining DNA to kind of flip. But if you have only one side that flips, then you break the DNA. And because you break the DNA, you end up with 2 segments, right? They're kind of duplicates of each other. Not really, but, you know, duplication is more in terms of the transposon itself. And you can also have the recombination caused by these transposons that try to move between chromosomes. Okay, so then you have shuffling between the chromosomes. So you can see how these mess up the DNA big time. And so you need kind of mechanisms to shut down this transposition, and the cells are able to do this. But some of the variability between our DNAs is caused by this. The L1, for example, has three mechanisms through which it causes changes in the expression of a gene. The first one is by inserting directly into the exon. Second one is by inserting in a place where it actually leads to the L1's anti-sense promoter being used to transcribe the exons, and so the transcription goes in the opposite side of the protein that is being messed up. Or the third one, if you have the L1 element inserting, let's say, between exon 3 and 4, so the exon, let's say, for example, the L1 element has a poly A tail, but you also have a poly A tail right here because of splicing variants and whatnot, then depending on which poly A tail you'll use during transcription to terminate your transcription, you'll end up having longer or shorter transcripts. And so this one, for example, could be inactive and this one active. And this is just because the L1 element itself has a poly A tail. So for example, when the RNA polymerase comes in and starts reading this DNA molecule here, when it gets here, it might use the poly A tail here, or it might use this one here. If it skips this one, you end up making potentially the full transcript, which is functional, but if it uses this one or whatever, then it messes it up, okay? Another important thing that the transposons led to is the shuffling of exons. So we've talked about proteins as modular in terms of domains that they have. And you'll see that a lot of proteins, they have combinations of different domains. So what happens is that when a transposon moves, sometimes it carries with it an exon from another protein. And so what happens in that case is that when you're going to transcribe, let's say, this gene here, which has an insertion of a transposon, then you'll be forming kind of this hybrid RNA molecule from the initial gene that you had, plus the exon that came with the transposon. Because the poly A tail will be at the end of the exon and at the end of the transposon. Depending on which one is being used to terminate transcription, you'll either stop here or you'll stop here. If you stop here, you'll be taking this with you, this exon 3 with you. If not, then you will not be taking it, okay? And if you take this exon 3 from this gene here, you make the hybrid RNA, then you reverse transcribe it to cDNA, and you insert inside another gene, then you'll end up having a new gene with 2 exons that were already there, plus one that you brought from the other gene. And this will lead to, let's say, for example, this is a DNA binding domain, the exon that codes for a DNA binding domain, and this gene here did not have any DNA binding domain. Well, now you have a kind of a chimeric protein that has a new function, okay? If you had one thing to remember is the poly A tail, because this is a little bit hard to imagine, but think about the poly A tail. Which one is being used, the one from the transposon or the one from the gene? An example of this is all these proteins, all these genes here. So all these genes here were generated through exon shuffling, and they were caused by basically translocation of different, or transposition of transposons. You can see that these genes have, I keep saying protein, but it's genes, have these domains, the LN domain, the G, and the B domain. And then depending on which protein, which gene you're dealing with, you will end up with a different combination of these domains, okay? This is all caused by transposition. Let's say, for example, in this case here, while transposition of the LN domain of this exon was carried here and here, and then you can see that in this case here, we don't have the LN domain, okay? Because this part here was not moved to these guys. But we have different copy numbers of the B domain, or the B exon. What else? Okay. So there is variation compared to earlier hominids, okay? There are many ALU elements and line or L1 elements that are different from humans, Denisovans, and Neanderthals, and these are very important to kind of evolutionarily placing us in time. There are 295 human-specific ones that are not found in the Denisovans and the Neanderthals. And then, obviously, there are differences between these two groups. You'll probably learn about them if you do a little bit of research following your paper that I gave you last lecture. You will see this kind of stuff. If you don't, that's okay. But these were very important in placing us in time compared to these two groups. Okay, our ancestors, let's say. Okay, last two slides, then I let you go. So let me just go like this. So in the selfish DNA, I talked about repeat sequences. So there are 3 types of selfish DNA. Selfish, remember, it's not as selfish as it's thought to be. So when you read it online, they say selfish, but it's not really selfish, okay? It actually has a purpose. We just talked about transposons. We talked about duplications in the other lectures. Now, the tandem repeats, we did mention them, but just to go back to them, there's 3 types. There's the satellite DNA, mini satellite DNA, and microsatellite DNA. If you remember, I talked about the satellite being close to centromeres, mini satellite being close to telomeres, so you can go back to the previous lectures. And the only difference between them really is kind of the size, okay? These are 100 to thousands of bases. Mini satellites can go up to 20 kb. Do you remember the hypervariable mini satellite that were used to detect differences between people? And
For the police departments and the
yeah, exactly. And then the microsatellites, which were used as molecular markers to kind of map genes on the genome, right? So these are dispersed throughout the chromosomes, and they're more evenly dispersed, so it's easier to use them as molecular markers than these 2 groups, okay? If you're interested in reading a little bit more about this, here's a list of them. Don't need to learn it by heart. It's just for you guys. Okay, that's it. Should I bring chocolate every time so that you don't sleep? Or it went like, he got chocolate and now he's back to boring. Any questions for me? No? Okay, so I'll see you next lecture. We'll talk about functional RNAs and answer your questions about the assignment if you have them. Okay? Take care.